Hello everyone, I'm Shunxin Wang from Shandong University. Today we are going to talk about meiotic recombination. At present, infertile couples account for about 15%. However, their successful treatment is less than 50%. Moreover, spontaneous abortions reach 10 to 15%, and birth defects are still high. Therefore, our human reproductive health is facing severe challenge. As we know, normal reproduction depends on the fusion of the sperm and egg. Sperm and egg are called gametes. Egg and sperm are haploid. They are fused to fertilized egg. The fertilized egg undergoes meiotic cell divisions and develops into a diploid human. However, how does a diploid organism generate haploid gametes? By meiosis. Meiosis is a specific cell division producing sperm and egg. A spermatocyte undergoes meiosis to generate 4 sperms, while an oocyte undergoes meiosis to generate 1 egg and 2 polar bodies. If meiosis is abnormal, it will lead to disastrous consequences. For humans, it will result in sterility, spontaneous abortions, and genetic birth defects. Among these, Down syndrome, that is 21 trisomy, is the most common birth defect in humans. The sperm or egg cannot be generated, or their quality are low, such as aberrant morphology and aneuploid chromosome numbers, which are leading causes for human infertility, spontaneous abortions, and birth defects. Egg and sperm are haploid, which have half of the parent genome. How do they accomplish it? By meiosis. During meiosis, 1 round of DNA replication is followed by 2 rounds of chromosome segregation. Homologs segregate at meiosis 1, and sisters segregate at meiosis 2. As a consequence, diploid progenitor cells generate haploid gametes. When we talk about meiosis, we have to mention mitosis. Mitosis is a type of cell division for the development of the zygote into an adult. During mitosis, DNA from generator cells replicates once to form 2 sisters. At anaphase, sisters are segregated to form 2 cells with the same genome complement as the progenitor cell. How do sisters know to move to opposite poles? In mitosis, 2 sisters are connected to each other, which will generate stable tension when molecules microtubules pull from opposite poles, and this tension will be also satisfied with checkpoint. As a result, 2 sisters are properly segregated into 2 poles. So, how do homologs know to move to opposite poles during meiosis? Similar to mitosis, homologs establish connections by forming crossovers, which ensure homologs' proper segregation. Crossover is the critical event of meiosis. It promotes chromosome proper segregate to generate normal gametes and keeps genetic stability. At the same time, it causes genetic exchange between parents' genomes and promotes genetic diversity. Crossover structure, that is chiasma, can be clearly observed at the diploiding stage of meiosis. Chiasma are composed of crossover at DNA level and cohesion between sister chromatid arms. Crossover numbers and distributions are tightly controlled. To ensure proper chromosome segregation, there is at least 1 crossover for per pair of homologs. When multiple crossovers exist on 1 chromosome, they tend to be evenly spaced, called crossover interference. It keeps crossovers far away each other, thus limiting crossover numbers. When crossover precursor numbers change, the crossover numbers tend to be kept constant, which is called crossover homeostasis. Abnormal crossover pattern can cause aneuploidy for a chromosome with a single crossover. Crossover position around the center of the chromosome arm are the optimal configuration, which will ensure proper chromosome segregate to generate normal gametes. However, single crossover located near the end of the chromosome arm, such as telomere or centromere, are suboptimal crossover configuration, which can lead to chromosome misaggregate to generate aneuploidy. Crossovers initiate from Meiotic programmed DNA double-strand breaks, that is DSB, which is mediated by sp11 complex. Under the role of MRX complex, DSB are processed into three-tail single-strand DNA with the help of recombination proteins, DMC1 and RID51. Three-tail single-strand DNA invades into homologous chromosomes to form D loops. A majority of D loops will develop into non-crossover by SDSA pathway, while only about 10% D loops will undergo DSBR pathway to develop into crossovers. Meiotic recombination occurs in the context of complex meiotic chromosomes. Meiotic chromosomes can be observed under electron microscope. We can see large loops, and their base are chromosome axes. Therefore, meiotic chromosomes are assembled into loop axis structures. Well-known axis components include top 2 mitotic and meiotic cohesions, condensations, and meiosis-specific axis components. After DNA replication, one chromosome consists of 2 sister chromatids. Their axes are conjoined, and the loops face to one direction. When synaptonemal complexes are formed between 2 homologs, synapsis occurs. Synaptonemal complexes are composed by different proteins. Although their consequences of amino acids are diverse in different organisms, the ultrastructure of synaptonemal complexes are conserved. During meiosis, chromosomes undergo a series of movements to recognize homologous chromosomes, do pairing, and synapsis. Their underlying basis is DNA recombination. Although meiosis is essential for the reproduction of both egg and sperm, it is highly sexual dimorphic. Female initiates meiosis in fetal gonad. After completion of recombination, oocytes enter the dictate stage and arrest as long as 10 to 40 years. At puberty of female, one oocyte resume meiosis and enter metaphase 2 each month and then arrest. It gets completed only when the egg is fertilized. However, in males, meiosis initiates from puberty, and it is a continuous process to form large numbers of sperms. In meiosis, chromosome segregation errors can lead to aneuploid gametes, which could occur between homologous chromosomes at meiosis 1, and also could occur between sister chromatids at meiosis 2. The fusion of aneuploid gametes will develop into aneuploid embryos. In humans, the frequency of embryo aneuploid is very high, can exceed 10%. Most of aneuploidy are spontaneously aborted. Those that survive are mostly trisomy 13, 18, 21, and X chromosome abnormalities, which is a leading cause of mental impairment and developmental disabilities. For example, Down syndrome. In chickling, about 90% aneuploidies derives from errors during meiosis. Furthermore, increasing maternal age is a powerful contributor to occurrence of aneuploidy. When mother is over 35 years old, the frequency of aneuploidy is dramatically increased. Compared to other species, human egg aneuploidy frequency is extraordinarily high, can reach 10 to 30%. By analysis, the patterns of crossover recombination are dramatically changes in these aneuploidy eggs. The frequency of chromosome absence of one crossover increases. The number of crossover decreases, and crossover distributions are altered. They are prone to located at the ends of the chromosomes. Whether meiotic crossover recombination in human oocytes are vulnerable to chromosome segregation errors. By comparing human male and female meiosis, we found that meiotic chromosome compaction is different between males and females. In males, the chromosome loops are larger and the chromosome axis is thus shorter. However, in females, the chromosome loops are smaller and the chromosome axis is thus longer. Since the female has longer chromosome axis, so it acquires more crossover precursors, more crossover destination sites, and thus more crossovers. Interestingly, even if female has more crossovers, but they have more chromosomes without even one crossover. Why? By investigation, we found that female-specific crossover maturation efficiency. In males, each crossover destination site will develop into a real crossover. But in females, some crossover destination sites cannot develop into real crossover, called crossover maturation efficiency. Crossover maturation efficiency alters crossover number and distribution. For chromosomes with a single crossover, crossover maturation efficiency can lead to lose the only crossover and thus result in chromosome missegregation. For chromosomes with two crossover, crossover maturation efficiency can lead to lose one crossover and thus change the crossover distribution, causing chromosome missegregation. Crossover maturation efficiency occurs at a fetal stage in human females, playing crucial roles before maternal age effect and determining the basal level of egg aneuploidy. With the maternal age increases, the degradation of cohesion maintaining chiasma structure further exaggerates chromosome missegregation, leading to more aneuploidy. Let's make a summary. Crossover ensures accurate chromosome segregation, which is essential for meiosis. Their numbers and distributions are tightly controlled, including obligatory crossover, crossover interference, and crossover homeostasis. Meiosis recombination occurs in the context of meiotic chromosomes, which are assembled into loop axes. We found female-specific crossover maturation efficiency is an underlying basis for high frequency of human female aneuploidy. That's all, thank you.